Hello everyone, welcome back to my channel. I'm Tepin and today we're going to learn how to submit protein coding gene in NCBI Bankit. So last time lots of comments were made in my previous video about submitting the protein coding sequences. So in this lesson I'm going to make it way more easier than before. When you don't have to do so much, you can just easily submit your sequence. So first you need a protein coding sequence and mine is this one. This is the protein coding sequence that I have. This is a translation elongation factor protein coding gene of a species of amino acid. So I'm going to use this as an example. So first you have what you have to do is blast your sequence. So after blast you will get something like this, like query cover percentage and all that. In here you have to click here alignment, and then it will come something like this. You will first find something like this. This query is your sequence and the subject is uh, the sequence to which it has been compared. For example, this is Felinus reverberinus. Uh, so this subject one is Felinus reverberinus and query is your sequence. So what we have to do here is click this CDS function. If you want to see some variation between your and the compare sequence, you can also do this pairwise with dots for identities. So it is also going to show where the sequence has been changed, all these red marks. So the most important thing that you need to pay attention to is this sequence. You can take either this or this or this or any one of these. Just you have to, for example, I will take this one. Okay, and let's copy it and let's just paste it here for now. I'm not going to do anything, just keep this. So this is it. I'm going to copy it. Sorry, I'm not going to copy. And let's copy this, our protein coding sequence. And we know that our protein coding sequence have these are the protein that it is going to code, and all this and all this, and then we have intron from here up to this point, and then again we have proteins, amino acid I mean, and then you have to do one thing, that is search ORF finder, and you'll find this one, NCBI ORF finder, and paste your sequence here, and then any sense colon, submit. So this will find open reading frame, and these are all different kind of open reading frame. So this one that I copied here, you have to look for this sequence specifically for each ORF. For example, in ORF6 you can see that there is no IIAAGTG. You can see there's no IIAAGTG. So I know that this is ORF7 is where I can find IIAAGTG. You can see. So this will give all the details that you need. For example, your strand is plus strand and your frame is reading frame is third reading frame, and note all this. You don't have to care about this one. Just note this too. The strand is plus strand, so your strand is plus strand and the reading frame is third. The third, yes. To confirm, you can also go other ORF. For example, here we have another uh, website where you can do this. That is ORF finder. Do the same thing here. Any codon, ORF reading frame, submit. You will find that all these things. You can see IIAGTG ORF number 1 in reading frame 3. You can see this is reading frame 3. Same thing you can do in many other websites we have here, but the best one is your ORF finder from NCBI. And then after noting all this is our plus strand and open reading frame is 3. You can just copy all this sequence. So I've already done this one. You can copy the remaining protein coding sequences that your protein codes for. You have to also keep in mind that this is not covering the entire protein coding. Protein, uh, I mean the amino acid that your sequence is uh, coding, because I will explain it. Let me just copy it for a second. Because specifically in my case, the sequence is partial. So what we can see here is that this is a partial sequence, and this is the query subject starting from 63. So this is the 63rd position of your sequence. You can say ATC ATT. Now ATC ATT. So it is actually showing you the results, the amino acid sequences from here up to certain point that we can find here. Uh, you can just copy all this, confirm the region by finding it here. So you can see this is the up to this point, this showing the amino acid sequences, and starting from here. So remaining these sequences, what about this? We don't know. So this is the disadvantage of this method. It's easier, you can submit it, you will get the SSN number. Those people who are working on proteins and they need specifically the amino acid sequences, they will need to work this region out by themselves. You can submit this in the GenBank and they will accept it because this is true. So now we have our protein coding sequence, and that is this portion. We can do one thing here, and then space. We'll replace the space with nothing. Then all sequence will be like this. This is the protein that has been that your sequence codes. So you can go to Bankit and start Bankit submission. Fill all your details. And in here you have to choose coding region and provide protein sequence data. Providing protein sequence data, you have to choose this one. Press add and you have to put the name of your protein coding parcel. So I have both 5' and 3' parcel. Describe your protein here, and here you can paste this in amino acid sequence. the protein that you got, and also other details that you know of. This showing plus strain and all this, intervals and all that. Don't have to do anything. Now you can continue and submit your sequence. So this is a very simple method. You just have to find first you have to blast, and you will get these all this sequence. You have to note some of this and go to ORF finder and then paste your sequence and try to find your ORF. In my case, this was ORF 7, which is a plus 10 and frame 3. And now that we know this, you can just copy all these things, not all this one, okay? These are introns. You have to select all this and then remove the gaps between them, then copy them and then submit it, like how I did it. So that's it. This is how you submit protein coding sequences in GenBank and NCBI Bankit. And if you have any more queries, please comment in the video, and I'll try to deal with them as soon as possible. And please be patient because these days I'm not getting so much time to make the videos, but I'll do it as soon as possible so that it can help you as soon as you need it. And that's it. Don't forget to hit like, share and subscribe, and we'll see you in the next video.